Hello, everyone, and welcome to today's webinar. Today's webinar is on transposable elements and epigenome evolution with Dr. Ting Wang. This event is part of the Summer Omics Learning Seminar Series, which is hosted by the M-LEEaD Center's Omics and Bioinformatics Core. M-LEEaD is the P53 Center at the University of Michigan. We had our first talk of the series in June just a few weeks ago with Dr. Betsy Foxman of the University of Michigan. And that event has been recorded and archived on our center website. And I will post the center website link into the chat later during this talk. In the interest of time, I'd like to get started. I'll just give the group a few logistical reminders. All participants are muted upon entry and for the duration of the event. This webinar will also be recorded and archived on the center website. If you have any questions during the talk, please submit them to the Q&A feature, which you'll see at the bottom of your screen, and you can submit those at any time during the event. I'd like to thank you all again for joining us today for this great talk with Dr. Wang, and I will hand it over to Dr. Maureen Sarter, the lead of our OBIC core.
Thanks very much, Katie, and good morning, everyone, or rather good afternoon now, depending on where you're at. Welcome to the second Zoom seminar of our Summer Omics Learning Seminar Series. As Katie said, it's being hosted by the M-LEEaD Center of University of Michigan, M-LEEaD being the Michigan Lifestage Environmental Exposures and Disease Center. I'm Maureen Sarter, lead of the Omics and Bioinformatics core, and I'm extremely pleased to be able to introduce Dr. Ting Wang from Washington University in St. Louis to you today. Ting is the inaugural Sanford C. and Karen P. Lowenthal, I don't know if I'm pronouncing that correctly, Distinguished Professor of Medicine at WashU. He received his undergraduate degree from Peking University in Beijing, his PhD in computational biology from Washington University, and then was a postdoc at UC Santa Cruz before then returning to Washington University as faculty in the Department of Genetics and the Center for Genome Sciences and Systems Biology. Dr. Wang is internationally recognized for his research on the evolution of mobile elements or transposable elements in our genome and their impact on gene regulation both in development and in cancer. His lab has developed the WashU epigenome browser, which some of you may be familiar with, which can be used to access many genomic data sets generated by several large consortia, including the NIH Roadmap Epigenome Project, ENCODE, the 4D Nucleome, and the NIEHS Target Project, which is how I've had the pleasure to get to know Ting. And so today, he will be speaking on transposable elements and epigenome evolution. So thank you so much, Ting, for presenting for us today, and I'll turn it over to you now. Oh, you're muted still, Ting. Still muted. Katie, can you, oh, there you go. Got it.
Oh, we're good, right? Okay.
Yep, now I can hear you. Thanks.
Thank you very much. Thank you, Maureen, for the very generous introduction. Thank you very much for having me here today in University of Michigan virtually. It's always a pleasure to visit the university here. I've long been wanting to come here and to see my old friends like Maureen, like Dana Dalinoy, also to meet new friends. I'm going to talk about transposable element, so I have to give a shout out to giants like John Moran, who's right here at University of Michigan, who's one of the reasons that I started working on transposable element. So I'm from St. Louis. St. Louis is right in the middle of the country. We're well known for this St. Louis Arch, which is the gateway to the Wild Wide West. Right? So we actually use this as a symbol for one of the projects that Marie mentioned, the WashU epigenome browser project. So we use this as a symbol, as a gateway to the human epigenome. So today I'm not gonna talk about the browser project. Rather, I'm gonna talk about something more nerdy, that is about transposable elements. So this is human genome, right? So we're genome scientists, right? So this is human genome. If you print them into book volumes, a lot what we do is to try to understand the meaning of the genome. Sometimes I get questions like, why you become a scientist? I think that's really because when I was a kid growing up, I really enjoyed reading books, right? So now, if you look at a human genome, there's so many books that we can read. As we all know, this is just one set of books. This is one reference human genome. Everybody's book is a little bit different from others. So everyone is a special edition. So in terms of number of books, this is endless. So for someone who enjoys reading books, I feel that, in genome science, I feel that I'm in heaven, right? So we also know that any book, right, even for the same set of books, there are many different ways of reading it, right? So there's skin cell has skin cell's way of reading a book, neuron has a neuron's way of reading this book. The same genome will express differently in different environment. So this is what we consider as epigenetics. In fact, also, epigenetics has been a hot area, a buzzing word over the past decade or so. When people talk about epigenetics, we often make some analogies, right? So being a computational biologist, one of the common analogy that we make is, the genome is like the hardware of a computer. Right? The epigenome is like the software of a computer. So you can run multiple software on top of this, the same hardware. So we use this as an analogy for epigenetics. Another way that I liked was reading a book, right? So different cells have different ways of reading a book. I'm going to give you one more analogy just to get the talk started. I really prepared this for my friend, Maureen and Dana, right? So I got this analogy of, what epigenetics is from watching sitcoms, right? So as Dana mentioned, I came from China. I got my undergraduate degree from Peking University. When I came to this country as a graduate student, I learned how to write some English, but in terms of, speaking and listening, it was basically zero, right? So now I can communicate. I still have a strong accent. I make grammar mistakes all the time, but we can communicate. But back then, when I sit in the classroom listening to the professor going like this, I could not understand a thing. Right? So, what do I do? Well, I have to bring a tape recorder to record the lecture and go back and listen to it like 6 times before I can figure out what the professor said. So one of the strategies, we're trying to solve that problem. One of the strategies is to watch a lot of TV. So back then I watched a lot of sitcoms, like, Everybody Loves Raymond, Mad About You, and of course, Friends. So I learned my English through watching TV, and also I learned a fair amount of American culture from there. In fact, I learned epigenetics from one of the episodes of Friends. So I think many of you have watched Friends, and you might remember this one episode where Joey Tribbiani, where he was wearing a man purse. Honestly, everybody loves Joey. I think he's a hero, right? So, men need purses, right? So we always have like a gigantic phone, and we have to stick in our pocket, and a string of keys, sticking our side. So if society allows us to wear a purse, I totally do it, right? Joey was a pioneer, so he started wearing this purse, right? And then he walked around and he was very proud of it, right? Of course, Chandler always poked fun of Joey, and Joey became upset. So Chandler goes, "Hey, what did I say? Why you're upset?" And Joey goes, "It's not what you said, it's the way you said it. It's not what you said, it's the way you said it. It's not the genes, is how the genes are expressed. That's epigenetics, right? So of course, this statement was repeatedly told to me by my wife on a daily basis. So that really got that concept of epigenetics imprinted in my brain, okay? So just this little joke prepared for my friends from sitcom friends, for my friend Maureen and Dana, all right? Okay, so we're gonna come back to our scientific topic. Before I get into transposable element, I want to squeeze in a recent project that we're leading that is called the Human Pangenome Project. As we mentioned, the Human Genome Assembly, that's essentially a book, a blueprint of humanity, but that genome reference is limited, right? So they're very, very useful, but they have limitations. One of the questions that we often talk about is, whose genome is it, right? You all use HT19, HT38, but whose genome is it, right? In fact, it's nobody's genome. It's actually a mosaic of sequences from just a few individuals who lived in Buffalo, New York when the genome project was initiated, right? So there's this new initiative that we're now building a human pangenome reference. The goal of this, so this project started September last year. The goal of this project is to really reinvent the human reference genome. What we're gonna do is to sequence 350 human genomes, telomere to telomere, haplotype resolved, make reference level human genome assembly from 350 individuals that represent human diversity. Right? So that's sort of a step number one. And step number two is we're going to come up with a better representation rather than a linear genome, but we're going to come up with a better representation to incorporate this genome diversity. So hopefully this project will really bring a lot of value to human biomedical sciences. Okay, so just a short advertisement of this new project. So what I'm going to focus today is to discuss with you about a human epigenome. In fact, the reference human epigenome project was one of the post-genome project that NIH launched and produced a lot of very useful resources. And this is a project that I was lucky enough to be part of. In fact, our Washoe epigenome browser was invented to host data generated in a human reference epigenome project. So the project ran from 2008 to 2015. We profiled the epigenome, which means DNA methylation, different types of histone modifications, open chromatin, gene expression, what have you, across multiple human tissues and cell types, also including ES cells and early development, right? So this project generated a massive amount of data, and it serves as a community resource for scientists to conduct their work. So the Roadmap Epigenomics project ended, but there are multiple community efforts that are continuing this effort, including the International Human Epigenome Consortium, the ENCODE Consortium, and the 4D Nucleome Consortium, which maps the three-dimensional structure of the genome. So creating yet another map that represents the three-dimensional structure. And of course, the Target project, where we're trying to map out epigenetic changes upon exposure. Right? So this is a community effort to generate resources, generate references for us to understand how the genome works. So we're generating a lot of data, but for me, the important part is the projects are actually generating data for part of the genome that I'm mostly interested in, but it's often less studied in the past 15 years of genomics research. That is transposable elements, right? So transposable elements actually make up at least 50% of the genome. Now, this is one of the first surprises after the human genome project was completed, right? 50% of the genome are known or can be recognized as derived from transposable elements like sines, lines, and LTRs. The other part of the genome that are, not recognized as derived from transposable element might simply because they're too old, right? When they're too old, we don't necessarily recognize their evolutionary origin, but if the part of the genome, our genome that are derived from transposable element is probably more than 50%. Some estimate this number to be 75%. Whereas coding exons, that's 1.5 to 2% of the genome. So the large majority of our genome actually made of transposable element. When we talk about the epigenome, we're only talking about DNA methylation in this context. So that means we're looking at CPGs. More than 50% of the CPGs of our genome are derived from transposable element. So over the past 15, 20 years, you've probably seen these three statements many times, right? So when genomics was booming, that was really because of technology. The first round was microarray, but whenever we did microarray analysis, we know probes are representing non-repetitive region, right? These transposon-derived sequences are often repetitive, therefore they're excluded from analysis from the very beginning. We don't even have data to begin with if we were using microarray. Short-read sequencing clearly took over the field. We often read like 60% of reads mapped to unique region and the rest discarded. Why do we discard them? Because they're difficult to handle, but also because we feel that, transposable elements are probably not useful. When you do computational work, for example, as a PhD student, I worked on motif finding. We'll always do analysis with repeat masked sequences because those repetitive region will screw up your statistics, right? So it's kind of ironic that, now my career is built on the part of the genome that I used to throw away as a graduate student. So why we throw away? Again, they're difficult to handle, but also because transposable elements are considered junk DNA. Many think they're useless. However, are they really junk DNA and useless? In fact, when transposable element was first discovered by Barbara McClintock, she actually called them controlling elements, right? Because they control gene regulation. 20 years later, Eric Davison and Royal Britain actually proposed that transposable element might help coordinate evolution of gene regulation, right? So Barbara McClintock won the Nobel Prize for the discovery of transposable element, but we actually believe she really deserves a second Nobel Prize for saying that this short piece of DNA can control gene regulation. That concept did win the Nobel Prize, but in a different context, right? So the idea is, these, gene regulatory network will evolve, and oftentimes this is built on the evolution of gene co-regulation. That means you want to have the same regulatory logic appearing multiple times that allow multiple genes to be coordinatedly regulated, right? So evolving the same logic multiple times can be difficult, but transposable element, they can regulate genes, they can make many copies, they're repetitive, so they become a really attractive source for developing this innovation in evolving gene co-regulation relationship. So that was the concept proposed by Eric Davison and Roy Brayton. So today, I'm gonna expand on that concept. I'm gonna argue that transposable elements are indeed full of energy. They're like the dark energy in the material world, right? But they're in our genome. I'm gonna make 3 points today. One is something that the field has really agreed upon after really a decade of effort, that transposable element really truly derive regulatory innovation. They help evolve gene regulatory network. But in the past a few years, we start to realize that they can also contribute to genetic robustness and epigenetic plasticity, right? So I'm gonna show you some evidence, use some anecdotes to illustrate some of the functions and roles transposable elements potentially play. So my lab actually spin around these three topics. We look at innovation of transposable elements' role in innovating gene regulatory network, but more recently we found their involvement in 3D genome as well as in the context of cancers. I'm going to use a few examples to illustrate these points. Right. So first, I'm going to quickly go over transposable elements and genome innovation. This is really used as a background because this is really what we have been working on over the past 10, 15 years. The concept is, what I have already described, that transposable elements are thought to be able to deposit the same or similar regulatory circuits multiple times, therefore coordinating the evolution of gene regulatory network. One of the first genome-wide evidence came from over a decade ago, and now in the context of studying a very important tumor suppressor gene, transcription factor P53. Right? P53 needs no introduction. It's the guardian of the genome. It's the most important tumor suppressor gene that is mutated in 50% of human cancers. It mainly works as a transcription factor, right? So what we found was in human, more than 30% of P53's genomic binding site were actually derived from a transposable element. This transposable element is an endogenous retrovirus. The virus actually entered the host genome about 60 million years ago. It spread it around for about 25 million years, and about 25 million years ago, this virus stopped spreading. So as a result, this 25, 30 million years of spreading resulted in 30% of P53's genomic binding site, right? So this is really a surprise, to me back then. P53 is so important, but there's so many binding sites of P53 were actually from a virus, right? So sometimes it makes you feel that, what are we? Are we human, or are we virus? Because there's way more viral sequences in our genome than what we consider human sequences. But in fact, these virus sequences are what make us human. So they're really deeply involved in shaping human-specific gene regulation. So P53 is not alone. This is not the only factor where transposable element contribute to its binding site. So we later showed that on average, 20% of transcription factor binding site in the human and mouse genome are contributed by transposable elements. So this is highly transcription factor specific. Here in this study, we looked at 26 pairs of transcription factor between human and mouse. We showed that sometimes transposable element contributes, really nothing to the binding sites of certain transcription factors. But in other cases, here CTCF, for example, more than 30% of CTCF's binding site in mouse were contributed by transposable elements. So clearly, this is a large impact on the host genome. More recently, using epigenomic data, we show that even though in an individual cell type, in individual tissue type, you often see transposable elements epigenetically silenced, but there are always some transposable elements that acquire active chromatin state, becoming promoters, enhancers, getting transcribed. If you look at across many tissue types, we found that somewhere around 20 to 25% of transposable elements can be in an active state in at least one of the tissue types that we profiled. Okay. So I think the field has really come to a consensus over the past decade or so. There are numerous studies coming out supporting the role of transposable element in contributing to the evolution of gene regulation. Right? So species-specific transposable element have indeed substantially altered the evolutionary divergent transcriptional circuitry of cellular processes. Right? So many groups have contributed to this. I think the field has come to a consensus as these transposable elements, they're not junk, right? So they can certainly contribute to gene regulation, and they have been doing so in a pretty substantial manner. Okay, so that's pretty cool, right? However, there's always something not quite satisfying. That is, till this day, right? Well, I guess, most of the time, right? Let's say, 99% of the studies that we've seen, we always see transposable element contribute to species-specific activities. Okay, that makes sense, right? Most of these transposable elements are species-specific, so they're innovators. They're making changes, right? So they're always innovating something, making some changes. Okay, so far we haven't seen anything that support that transposable element can contribute to a conserved function. As we know, conservation is an important measurement of functional significance. So we've been always sort of dissatisfied, not finding evidence of transposable element contributing to conservation until we looked into 3D genome. Okay, so this is a study that we're doing in the context of the 4D nucleome project. So a graduate student in the lab, Mayank, is very interested in understanding contribution of transposable element to the 3D genome structure. 3D genome structure is gaining a lot of tractions in the field over the past few years, again, due to technology development, right? So as we know, DNA is like 2 meters long. It got wrapped around and packed in this teeny tiny nuclei. So this process, when I was in school, I was taught that this is essentially a zip process, right? So it's really compacting. But now we know this structure has intimate relationship to gene regulation, right? So DNA is wrapped around the nucleome. They form chromatin domains, topologically associated domains, compartments, and all the way to this chromosome territory. There's like different levels of structures, and this is a highly, highly regulated process, had everything to do with gene regulation. So these are the typical things that we look at when we study chromatin interaction. So in the context of today's discussion, we're mostly focusing on topologically associated domain and chromatin loops. The experiment that we often do is so-called Hi-C, which generates this contact frequency map, right? So this is a heat map indicating the likelihood frequency of interaction between two regions in the genome that are far away in linear genome, let's say this spot and this spot over here, but in the 3D genome, they're contacting each other, represented by this increased intensity of this little square here, indicating these two regions are touching each other in the 3D structure. So from a Hi-C map, you can sort of derive a schematic of how the chromatins loop and interact with each other. The reason that we're interested in this is because these structures are often held together by architecture proteins, including CTCF. In fact, CTCF is one of the most important architecture protein holding this chromatin loops together and sitting at the boundary between topological associated domains, okay? So the structure that we're talking about are these chromatin loops, and at the anchoring site, you often see architecture proteins, including CTCF. So in the early days, we already see that 3D structure, 3D genome structure can be conserved, right? So this is an early study published by Bing Ren's group showing a comparison between human and mouse in terms of their three-dimensional structure, right? So you don't need to be a like a computational biologist to see that this is actually significantly related, right? So the chromatin domain structures are highly conserved. The boundaries are highly conserved. Okay. In fact, we estimated about 50% of the human loops actually are conserved. That means we can find a corresponding loop in the mouse lineage, right? But that also means 50%, the other 50% of loops are species specific. Okay, so half of the loops are conserved, half of the loop are species specific. Of course, this is limited by the specific cell type that we looked at, the specific technology, right? But by and large, we see this picture. Now I'm going to bring back CTCF from this early study where we found, actually not just us, many people have actually seen that a lot of CTCF sites are in transposable elements. We also know these transposable elements are species specific. Therefore, it's very, very natural to hypothesize that these species specific transposable elements are important in mediating species specific loops, right? So that's very intuitive, and that's that sort of belongs to this concept of innovation, right? Transposable element can innovate gene regulation. In this case, they're innovating the 3D genome. So that's pretty easy to think about. Okay, so we looked into that, but we also looked at conservation. I think yes, transposable element can drive innovation in 3D genome architecture, but can they contribute to the conservation of 3D genome? So that led to some very interesting discoveries. So first of all, let's look at this, just some overall numbers. 50% of the loops are species specific, in other words, they're not conserved, right? Among those, about 20% of them are derived from transposable element, right? So this is very consistent with some of the early studies that we've done. 30% of P53 binding sites are from transposon. On average, 20% transcription factor binding sites are from transposon. 20 to 25% transposable elements can adopt an active state, right? So we're seeing this just as we expected. So the unexpected finding comes from looking at the other 50%, that is the conserved chromatin loops. We found that 10% of these conserved loops are actually derived from transposable elements, right? So this is a little bit counterintuitive because transposable elements are species specific. How can they derive conserved binding loops? Turns out, this is a very, very simple principle. This is something that we've known for many, many years. That is called transcription factor binding site turnover, okay? So what happened in the course of evolution is, let's say this is an important chromatin boundary, okay? Between human and mouse is conserved. For whatever reason, they're conserved. That's being held together by a conserved CTCF site. So now when you have transposable element jumping into the genome, some of the transposable element actually carry CTCF binding site. We have some analysis shown that these binding site, these transposable elements are methylated, but sometimes they become demethylated, which expose these CTCF sites. So CTCF is a methylation sensitive DNA binding protein, right? So when these binding sites become exposed, okay, they can actually be used to hold the same chromatin loop, which gives an opportunity for the original binding site to mutate, right? So here two things can happen because the pressure of maintaining this binding site is essentially gone, right? So you can use this binding site or you can use this binding site. Oftentimes transposable elements got away, right? But sometimes these transposable elements get fixed. So there's mutation happening on the original binding site, and these transposable element took over and become the new anchor site of this essentially the same chromatin loop. Right? So this is actually a classic transcription factor binding site turnover event. Right? But in this case, we're seeing this because we're looking at 3D chromatin structure. So this is a conserved function mediated by non-conserved family of sequences, right? These species-specific sequences can actually take over and maintain chromatin structure. I'm going to just show you a couple of examples here. So here is you're looking at a human and mouse comparison. These are conserved chromatin domains. So we're looking at the specific boundary between this domain. Okay. So in human genome, we see a very nice CTCF binding peak. This is a ChIP-seq on CTCF, and we see a very nice looking CTCF binding site, right? But if you look at a aligned mouse sequence, we see that this sequence, this part of the sequence has decayed. Right? However, if you look at mouse genome, about 600 base pair away, there is a B3 element insertion, which this B3 element contains a perfect looking CTCF binding site, and this B3 is bound by CTCF. So this is the ChIP-seq peak. So the hypothesis is this B3 element insertion actually replaced the function of the original CTCF binding site. So this binding site now are free to mutate, which it did, and this B3 element took over the function. Okay, so we performed some experimental validation. So in this case, we're going after a transposable element called L1 M3F, which serves as a boundary element between this chromatin domain and this chromatin domain. So the red arrow represents this transposable element, right? So here we used CRISPR to delete this transposable element, therefore removed the CTCF binding site, right? What we found is now these two chromatin domains start to merge, right? So here, this CTCF, this transposable element actually forms 3 loops with these 3 points. Once you delete it, the loops are gone and the domains start to collapse. So over here, this is a measurement of interactions. So before deletion, we see there's very little cross-domain. There's actually very little cross-domain interactions, but once we deleted the transposable element, the inter-domain interaction increased to 3-fold, right? So this demonstrated that these transposable elements are actually really important for holding these 2 chromatin domains together. So these are control domains, right? So this is one example. We always know, like, one example isn't enough, right? So you need to show this multiple times. So we went after another example. So in this case, there's a transposable element L41 derived loop anchor or domain boundary here. We deleted it. We did Hi-C and tried to see if the domain structure changed. Only in this case, the domain structures did not change. Well, we will have like true positive, false positive. Not every example will work. So sometimes, you have the urge of just publishing the other one and then leave this alone, right? No, that's not what Mayank wanted to do. So he thought, you know what, Ting, give me more money. I'm going to sequence more. I'm going to generate an even higher resolution map. The original map was done at like 100 KB resolution. So he sequenced a little bit more and it generated a 1 KB resolution map for this region. And now something interesting emerged, right? Turns out, when you deleted this transposable element, right? So another CTCF site that is about 30 KB away actually becomes new boundary. So the other side pick up, right? So if you look at a map of 50 KB or 100 KB resolution, these two cannot be distinguished, right? So you are not going to see change of chromatin domain. But with a high resolution map, you see that the first boundary is actually gone, but the boundary shifted about 30 KB. So the large structure between these 2 chromatin loops, chromatin domains, these 2 topologically associated domains are maintained, right? So basically, if you take out the first binding site, which is derived from transposable element, right? Another site actually pick up. What's interesting is the second site, right? So the site that picked up over here, represented by D, this is actually the ancestral site. in other words, this is the site that human and mouse shared, okay? So what happened here is we essentially reversed evolution, right? So in the course of evolution, we believe that human mouse started, using this D altogether, right? But in the human lineage, this LTR41 inserted a new CTCF binding site. Now, given, time during the course of evolution, this CTCF site took over and become the dominant CTCF site that maintain the boundary between these two chromatin domains. For whatever reason, these two domains need to be retained, we don't know, but this demonstrates that this transposable element is now maintaining this domain. Once you deleted it, the domain shift back to the ancestral state. Okay, so this is an example where we see a local turnover of chromatin boundary mediated by transcription by transposable elements. So I'm not going to show this part of the data, but some new analysis demonstrate that there are really two modes of replacement depending on the orientation of the transposon insertion. Okay. All right, I'm gonna now switch gear to talk about transposable element and epigenetic plasticity. I'm gonna describe this in the context of cancer. Okay. So, talking about epigenetics at University of Michigan, I can't give this talk without showing the agouti mice, right? So this is what Dana worked on and really changed the field. Now look at these two lovely animals, right? So you almost want to snuggle with them, right? So these two are very lovely, but obviously they have very different phenotype. The important part is they actually have exactly the same genotype, right? So every base in their genomes are identical. The difference between their phenotype is because of their mother's diet when the mother was pregnant, right? So the mother of this animal just ate normal mouse chow, but the mother of this animal, in addition to normal food, she also took in vitamin supplements, including folic acid and B12, which serves as muscle donor, right? So the molecular mechanism behind this phenotype is actually mapped to a transposable element, right? So the causal gene is agouti gene. About 100 kb upstream agouti gene, there's a transposable element insertion called IAP. Now this IAP element can serve as a cryptid promoter and drive overexpression of agouti gene at the time and in tissue where this gene should not be expressed. Right? So that results in all the bad phenotype. When the mother takes muscle donor supplements, the baby can actually methylate this AP element. When the element is methylated and repressed, the agouti gene expression returned to normal, right? So you ended up with a normal looking project. Now, this is a classic example. This is really the example that got myself into the field of epigenetics back in 2000, I think 6, 7, 8-ish. Okay, this is the reason. Now, one of the reasons is, as we know, there are so many transposable elements in the genome, 50%, right? So there are so many opportunities for the regulation to go wrong, and then that's gonna contribute to dysregulation of genes and potentially contribute to disease. So how many human diseases can be caused by this mechanism, right? So we essentially started a project to look for the human agouti, right? So we want to find a human disease where the mechanism of disease is really like the agouti mice, where a transposable element lost the DNA methylation and became a cryptic promoter and drive abnormal overexpression of a human gene. So we started looking for such diseases. So where do we look, right? The first place that we started looking is very obvious, is cancer, right? Because we know there are a lot of abnormal activity in cancer. We know cancer's genome undergo epigenetic reprogramming or deregulation. lots of transposable elements are losing demethylation, so we feel that we have a bigger chance of finding something there. Now, what type of gene do we look for? We look for oncogene, right? We know cancer cells will activate their oncogene, will overexpressing their oncogene. In fact, that's written in the textbook, right? So many years of cancer research have shown that there are 3 genetic mechanisms for activating oncogenes. There's mutation, there's amplification, and there's translocation, right? So we're really looking for a force mechanism that is epigenetic in nature that is causing oncogene upregulation. So typically, you have oncogene in the genome. They have low resolution, low expression. There are a lot of transposable elements around. They're epigenetically silenced, so everything's cool, right? But during tumor development, right? So there's epigenetic insult. There's misregulation. Sometimes we see transposable elements losing demethylation, losing epigenetic control. Sometimes that's going to result in transcription, and that transcription can go into nearby oncogenes, just like IAP element going to a agouti gene. That's going to result in an oncogene overexpression. Now, this is cancer epigenome evolution, right? So what we're looking for is actually very simple in the sense that, the cancer needs to evolve. The cancer cell can evolve genetically, you can mutate, you can amplify oncogene, but you can also try to find a new promoter for this oncogene, right? And here, transposable element, we know they're potent, they're regulatory, right? They have regulatory activity. So they provide a resource for these oncogenes to pick up as a new promoter, which help them overexpress genes that's going to be beneficial for cancer cells to grow, right? So that's the event that we set out to look for. We looked into TCGA data, where there are a lot of RNA-seq data. So here we developed our bioinformatics infrastructure to specifically look for those events, right? So RNA-seq data, typically people map to genes and find gene expression level. But in this case, we specifically looked into transposon and gene connection, right? So in this case. So we found that this is actually pretty widely spread, right? So we look at many cancer types that TCGA profiled. At the end of the day, we found that at least 50% of the cases, right, where we can find an event that we call oncoexaptation. In other words, in these cases where the cancer itself, the tumor is highly expressing an oncogene, but that expression does not from its own promoter. Rather, the expression starts from a transposable element nearby, right? So this is what we call an oncoexaptation event, and this happens in at least 50% of the tumors that we looked at, okay? The reason I say this at least 50% is because, this study is based on RNA-seq. As we know, many RNA-seq data are 3' biased. So in this case, we really need to see the 5' end of the transcript to be sure. So if we look at what kind of element, what kind of transposable element contribute to this type of phenomenon more, and we see LTR winning across the board. So this is also consistent with previous knowledge that LTR is contributing more regulatory elements in the genome, but obviously line and cyan DNA element, they all contribute to a certain degree. So I'm going to just go over one quick example here. So these are some of the top candidates. Basically these are oncogenes, well-characterized oncogenes, whose expression are not from their own promoter, but rather from a transposable element, which is the first term here. So let's just look at this one at the bottom, right? AlloJB Lin20AB. So in this case, Lin20AB is the oncogene. It's a bona fide oncogene. It regulates the LET7 pathway. What we found is in many cancers, right? So this is a variety of cancers. In many cancers, the expression of Lin20AB indeed is very high, right? But the transcription does not start from its own promoter. Rather, the transcription starts from the AlloJB element about 20 kb upstream, right? transcription starts from here. It actually goes all the way into lin28B, resulting in the high expression level of this oncogene. Okay, so what's the relevance of this, right? So at the minimum, this is actually a very interesting prognostic marker. So what we found is in patients where we see this type of lin28 expression, which means lin28 driven by allo-JB. If we see this type of lin28B expression, these patients feature much worse than those that without allo-JB driven lin28B expression, right? This is actually easy to explain because these patients usually have much higher lin28B expression, right? When you have a higher oncogene expression, the tumors are more aggressive, right? So therefore, these patients feature much worse. So we decided to look at the epigenome of these cancers. Right? So over here, this is this part of the genome. This is the lin28B promoter, right? So what we're looking at here are two lung cancer cell lines, and the specific technology we're showing here is called CAGE. CAGE allows us to grab the 5' end of the transcript or the transcription start site, right? So what we're seeing here is in this cell line, H1299, the transcription does not start from lin28 promoter, rather it starts from over here, right? So transcription goes all the way into lin28B. We actually also validate this with PacBio long-read sequencing. Now we look at the epigenome, we look at DNA methylation. What we see here is this region where the CAGE peak locate in H1299. This is completely unmethylated, right? So these little blue bars represents methylation level. So this is completely unmethylated. Whereas another cell line, this is sort of half methylated, right? So this region exhibit bona fide promoter epigenetic profile. So what we're looking at here are CAGE-seq peak, CAGE-seq H3K27 acetylation, K4 monomethylation, and K4 trimethylation, K9 acetylation. So this is classic promoter marks. And if you look at where the signal comes from, it comes from a allo-JB element, right? So we also did a series of experiments to show that this is indeed the element that gives the promoter activity. But if you look at the normal tissue, so this is a panel of normal tissue across different tissue types, like heart, pancreas, and whatnot, and you see this region is completely methylated. This is consistent with our general knowledge. This is actually a repeat-rich region. So this allo-JB element is methylated, right? So this is as boring as Midwest, I don't know. Anyway, so that shows that this allo-JB, at least in this tumor cell line, exhibit very active promoter marks. The other thing that's really interesting is, so if the transcription starts from allo-JB, this transcript actually gives us a slightly different protein, okay? So this is the conventional lin28 protein, right? But the transcript starting from allo-JB actually removed 3 amino acids from the very end terminal and added 22 amino acids, right? So these amino acid sequences were contributed by the transposon sequences, okay? Now, we do not believe that this altered protein's function. We actually did rescue experiment to demonstrate that. But this actually allows us to separate these two types of protein, these two versions of lin28B on a Western blot because their size is slightly different. So here we're looking at 4 cell lines, right? These 2 cell lines where we predict we're gonna get this allo-JB driven lin28B, whereas these 2 cell lines are expressing the conventional lin28B. When we run a Western blot, we see this size difference, right? So this is the conventional lin28B, and this is the lin28B that is driven by a transposable element, and then it added 22 amino acid to the N terminals, okay? turns out that the field has been studying this version of Lin20B for over a decade. Okay.
Hello,
All
Ting.
right. Oh, yes.
I just wanted to check in and let you know we're almost approaching the end of our time, in case you want to wrap up, and thank you.
I'm sorry, I lost track of time. Anyway, so I'm gonna quickly go over this. So the next set of experiment really shows how we confirm that AlloJB is the causal factor. So throughout, we did two types of experiment. We did CRISPR knockout. So in this set of experiment, we deleted AlloJB, and we showed that Lin20B actually indeed disappeared, right? So we carried this experiment to nude mice and show that when you delete Lin20B promoter, the cancer cells can still form tumor. But if you delete AlloJB, the cells do not form tumors anymore. The one last experiment we did was to show that Lin20B is under epigenetic control, right? So on the left, we show that we can target Lin20B for methylation, which reduces transcription of Lin20B. We can target AlloJB for methylation, right, and then reduce Lin20B expression. On the right-hand side, we show that we can target AlloJB for site-specific demethylation and trigger the expression of this Lin20B protein. Okay, so that's sort of the last experiment that I want to show. But this really reveal a very interesting future direction about epigenetic therapy. Okay, because what we showed here is by epigenetically manipulating the cancer genome, you can actually trigger the production of this type of Lin20B, which has 22 amino acids added to the N-terminus. Now, this is, again, not unique just to Lin20B. When you have a Lin20B with 22 new amino acids appended to the N-terminal, imagine this is not an oncogene. This is just some dummy genes, dummy protein there. This 22 amino acid is something that the cancer cell have never seen before, right? Therefore, they can serve as new antigen, right? So this experiment actually opens up a door for us to explore the potential of transposable element in driving chimeric transcript that can potentially produce novel peptide for us to target, right? So this is sort of a summarize our current effort and thoughts, right? So we started with a genome where transposable element is demethylated, methylated, right? Sometimes in cancer, we see this demethylation that drive oncogenic, but sometimes we can use this information, we can use this capacity to drive production of chimeric peptide that can serve as a potential therapeutic target, okay? So to summarize, so on this part, I showed some evidence of oncogenes being upregulated by epigenetically reactivated transposable elements. I showed that we can manipulate junk DNA genetically and epigenetically and result in a cancer-relevant phenotype. And finally, we showed that this chimeric TE gene fusion transcript, they can serve as a rich resource for new antigens, right? Just to sum up, so today I hope I've shared with you some of our thoughts about this transposable element, really the dark matter, dark energy in our genome. We showed that over the past decade or so, the field have come together to realize the regulatory innovation transposable element has contributed to gene regulatory network, but more recently we start to see that transposable element contribute to genetic robustness as well as epigenetic plasticity, so that it allows phenotypes to evolve without genetic event, right? So the evolution happens at epigenetic level. Finally, I'd like to give acknowledgement. I'm very lucky to have recruited a group of crazy individuals to work together. So these are lab members. So I just want to say that in order to make this picture, this is a very nice looking picture from last year's summer party. Unfortunately, we won't be able to have one this year. I just want to say that, all the work that I showed are done by them, but there are a lot of work behind the scene, just like taking this picture. This is a nice picture, but in order to take a picture, they actually dropped me on the floor like 4 times. I'm sure they did it on purpose. So these are the collaborators that I've had over the years. So my lab works a lot in the context of different consortiums. So we're gonna, and these are the collaborators on the cancer side. Okay, I always want to argue that we should put Barbara McClintock's picture on a $20 bill. We'll continue fighting for that. Okay. Thank you very much. I'm sorry that I lost the track of time.
Thank you, Ting, for that really interesting seminar. It is, and it looks like you have a really fun lab as well. It is a couple minutes after 1 o'clock, but for those of you who can stick around, we can take some questions now. So I'll remind you that there is the Q&A part of the Zoom session that you can type in your questions. So you can go ahead and do that now, and then we'll ask the questions to Ting. I guess to get us started while anyone else is typing in their question, I will ask one. It was really interesting talk and made me think about, TE elements and epigenetics in a somewhat different way. And, so we can think about each new transposable element when it's first introduced to our genome as being, wanted or unwanted, or useful or not useful to our genome, and may potentially leading to natural selection. So I was wondering whether it's known or if there's any evidence that TEs that have higher sequence similarity to one or more of our transcription factor binding motifs, if they tend to be more expanded in our genome than others.
Thank you for a very interesting and important question. This is really talking about, which comes first, right? Chicken or egg, which comes first, right? So we believe that some of the transform factors are, we call them master regulators these days, like P53, right? So some of these transform factors, they became master regulator because potentially some transposable element has been helping them depositing transform factor binding site. So indeed, in this case, I think sequence is very important. These motifs are indeed very, very important. Sometimes it's beneficial for the viruses to have a motif of a transform factor binding site, have the binding site of a transform factor to help its own proposition, right? But sometimes it is indeed because of their activity, because they deposit many, many sites across the genome that actually give the transform factor an opportunity to recruit more target genes and grow its regulatory network. So I personally believe that it actually goes both ways, right? So not only that viruses take advantage of host factors to promote their own transcription, right? But also they deposit more binding site that actually helped transform factors to grow a bigger regime.
Great, thank you. I think you froze for a second there, or I did. Anyway, so we have one question up now. Someone is asking, how do TEs differ from non-regulatory regions?
Well, I think they're the same thing. So TEs are deposited across the genome. I'm not going to say, in fact, I don't believe that all TEs are regulatory, right? So there are sequences there, right? Some has some sequence, some TEs contain sequences that have regulatory potential. That doesn't mean that they come in with a purpose. It doesn't mean that they come in to help us. It doesn't, I certainly do not mean that all TEs are functional, right? So by and large, they are depleted of, regulatory sequences, but because their sheer volume, because they occupy such a large space, occasionally we see some transposable element getting adapted, domesticated, serve a domestic host function. So when we say non-regulatory region, it really depends on how we define that non-regulatory sequence. I would say large majority of TE sequences are indeed non-regulatory. Okay, some of them, become regulatory because, the host somehow domesticated them for their own purpose, for the host's purpose. So they're really not difference, in our genome. They're equal. They're just sequences, right? But they're sort of the reservoirs for gene regulation, for evolution to act upon.
Okay, there aren't any other questions. Oh, someone said, thank you for a wonderful talk. Is there a bias towards the TEs seeding sites of transcription factors of viral origin or homologous to viral factors?
So, sorry, can you repeat? There was a glitch.
Oh, yeah. Is there a bias towards transposable elements seeding sites of transcription factors of viral origin or homologous to viral factors?
I don't think I've looked into this before. I don't know I don't know the answer to this question. Yeah.
Okay, well, at this time, I don't see any other questions. Katie, can you jump in and make announcements for how to move over to the other discussion?
Of course, yeah. Thank you again, Ting, and thank you everyone for joining us today. Just as a quick reminder, we will have the next Soul series lecture on August 10th. I'm going to post a link in the chat box for everyone who's still here. And then if you are joining us for the discussion, and we'll start it right now. I've sent the details to those of you who are invited to your email this morning. If you have any questions, please feel free to email me. And thank you again.
Thank you.